Now, this is the gene tag, quite complicated topics. Okay, so to warn you guys first, for these topics, I may need about 3 weeks. Okay, so that's why this week I have to call you guys back for the class. Okay, because I need about 3 weeks. Okay, to complete these topics. Gene tag and gene tags. Whatever we learn is just a model. Means that we use insulins as a model, but in the real exam, you can see that we may have a lot of the different type of the question. So it's one of the most difficult topic in A2. Okay, so you have to grasp the concept correctly, then you will be able to proceed to the application. Okay, so what are our learning outcome for today? We may not be able to complete everything within the 2 hours, okay, for today. So let us look at what are the learning outcomes that we need to know here. So first, we need to define the term recombinant DNA. What does mean a recombinant DNA? So sometimes we do write it as an rDNA. The R stands for recombinant DNA. It's still the DNA, but what is meant by recombinance? And then we need to explain the genetic engineering involving the extractions of the gene from one species, or we synthesize the genes in order to place them in another organism. So basically here involve what we call the gene transfer. But this gene transfer is not because of the sexual reproduction, it's not because of the bacterial conjugations, but more to the man-made process. Basically, we take one, we cut, okay, the DNA, a particular DNA, particular gene from one species, and we transfer to another species, okay? So that the receiving organism now can express the gene products. Okay, for example, okay, what we're going to learn here is the insulin gene. So we do know that bacteria, they can't produce insulin because they don't have the insulin gene. But if we want the bacteria to produce the insulin genes, insulin for us, the gene product insulins for us, then we have no choice but to transfer the human insulin gene into the bacteria. And to do this, we need a certain tools here, right? We need a tools. For example, if let's say I ask you to cut a paper, to fold a paper, to cut the paper, the shape, and then paste it on another paper. So you need a cutter, right? You need the glue, crayon. So in this case, what we're going to look at it, what are the genetics technology tools that we need to use here? Basically, first is called restriction endonucleases, and commonly known as a restriction enzyme, or we call it RE, okay, restriction enzyme. We do involve this, what we call the reverse transcriptase, what we have learned before in HIV, okay, reverse transcriptase. So we do need to know why we need to use a reverse transcriptase, and we need another item here called ligases. So if you analogy with, let's say, for example, I ask you, okay, cut the blue paper and then paste it on the white paper. So restriction enzyme basically is a cutter to cut. Ligase basically is the one we use it to paste. So in this process, we involve cut and paste. So the cutter here is the restriction enzyme, ligases is the one to paste. Reverse transcriptase, I won't talk in the detail at this moment. So later, we're going to look at it. Okay. Now, the thing here is, if we take the human genes and then we want to paste it in the bacteria, then we need someone to transport this, right? For example, we do learn in the infectious disease, malaria, right? So someone have to transfer the plasmodium, the Anopheles mosquito. So in this case, who help us to transfer, right? Who help us to transfer the gene of interest, insulin gene, into the bacteria? Then we need to depend on what we call the vector. Okay, plasmid is what we are going to focus. So we call them as a molecular vector. The one that help us to transport or to transfer the gene of interest into the bacteria. Okay, and we do know that we need to know why we need to include promoter sometime and some other control sequences that we add in. Okay, so that the bacteria or the cells can help us to produce insulin. And last, we need to know whether successful or not this bacteria already take in. So we look at genes for fluorescence or easily stained substance as the marker. So this marker we call as a genetic marker. So this genetic marker is very important to tell us, oh, success or not. If no, then we're going to culture something that not success. okay, let's say, or the bacteria that didn't take up the gene of interest, then what for we want to culture them. Okay, so this is basically the process of what we call the gene transfer. Okay, so we started with the definitions, what it means by genetic technology. So it's sometimes we also call it as a genetic engineering or recombinant DNA technology. Okay, so recombinant here, what's the idea? What is mean recombinant? Recombinant means recombine. What we do here, very simple, we combine the DNA from two different species. Okay, so if you go back to, okay, you recall back the previous lessons, we talked about the reproductive isolation. When talk about reproductive isolation means that when two different species of organism, when they interbreed, then you can see that they may not be able to produce fertile offspring. Okay, so that's why to overcome this problem, we don't let them interbreed. Instead of let them interbreed, we transfer particular DNA, particular gene from one organism, pass it to another organism. So even though we can do it between two different species, but we won't cause the offspring to be infertile. Basically, that has nothing to do with the interbreeding. Okay, so what is the idea? Why we want to do this? Basically, we want to manipulate or modify the gene so that we can improve the products or improve the methods. So the whole process may involve the removal of the gene from one organism, transfer it to another, so that the particular organism will express the proteins for us. Okay. So what it means by recombinant DNA? Recombinant DNA basically involve the DNA that contain the length of the nucleotides from at least two different organisms. So this DNA molecule is known as recombinant DNA. Are you clear? As long as it consists of two different organisms, two different species. Okay, so recombinant DNA is a DNA made by joining pieces from two or more different sources. And this is very important because we can overcome the barrier to gene transfer between the species, because we do know that between two different species, if they interbreed, they always will have what we call the reproductive isolations. So for those who are confused about reproductive isolations, later you can go back to the link, okay, that I share in your WhatsApp group to check, okay, to check on what is meant by the reproductive isolations. And in this case, we only transfer the gene of interest, the genes that what we want, so always involve a single gene. So in this case, we can transfer between different kingdom, even from the bacteria to the plants, from the humans to bacteria, even you cross the kingdom, you cross, okay, the domains. Okay, so this we're going to look at it in the next topic when we talk about this biodiversity. Okay, so what are the tools we use in the genetic engineering? So we have the cutter here to cut restrictions endonuclease or restriction enzyme. So we need the transporter here, vector, okay, or the transporter, or sometimes we also call it as a vehicle, to transport gene of interest into the host organism. Okay, so but in order to do this, we need to paste it, right? So cut and paste. So our glue here is a DNA ligase, and we need the host organism to produce the gene products for us. Okay, so reverse transcriptase. Now, from the word transcription, we do know that transcription actually use DNA as a template to form mRNA, right? So this is what we call the transcription process. So the reverse way is known as the reverse transcriptase. So means that use mRNA as a template to make a DNA. This is the reverse transcriptase job, okay, the role. So basically, we're going to synthesize DNA or complementary DNA, the cDNA from, okay, from where? From mRNA. So this is the role of reverse transcriptase. Okay, so don't worry, at this moment, we're going to go in one by one. We're going to go in one by one. Okay, so far, do you guys have any questions at this moment? You can type out your question in the chat, Zoom chat there. Okay, you can private message me, it doesn't matter. Okay, I can see it. Okay, so if no questions, then I continue to this restrictions endonucleases, the cutter here. Okay, so I won't go into a very detail about these restriction endonucleases or restriction enzyme. Basically, they are a protective mechanism by the bacteria to cutting out the foreign DNA. Let's say, for example, okay, when they are infected, okay, when they are infected by the viruses. So viruses, if you look at how the HIV reproduce, they actually release their own genome into the bacteria or host cells, and then they're going to interrupt our DNA, interrupt the cellular functions. So in order to protect bacteria, okay, bacteria, they don't have immune system. What they do have this restrictions enzyme. So this restriction enzyme can help them do what? To chop down, okay, to cut down, okay, to cleave at the viral DNA, so that viral DNA cannot give instructions to the bacteria and use the bacterial protein synthesis mechanism to produce viral proteins. Okay, so what they do, they actually cut out, okay, cutting out their foreign DNA. Okay, and we already discovered hundreds of different kinds of this restriction enzyme, and we realized that they are very specific. So this enzyme can recognize a particular site of the DNA. For example, okay, one of these enzymes can check on GAA-TTC. Okay, the reverse one, GAA-TTC. So whenever this restriction enzyme check on this, when they discover, oh, got this sequence GAA-TTC, then they're going to cut it, cleave the DNA. So it means that no longer, your DNA no longer intact. You can see that very long, right? So they're going to check one by one, until they check on this. Oh, so found GAA-TTC. So when they found GAA-TTC, what they're going to do, they're going to cleave this DNA, make it become fragments. Okay, and the recognitions here, when they can recognize this site, so this site is known as the restriction site. Are you clear? The word restriction site. So once they recognize this site, they're going to cleave it so that the both DNA strands now can be separated. So this site is known as the restriction site, normally 4 to 6 nucleotide long. So in this case, you can see that, okay, they have 6 nucleotides long, and most of the times they have this what we call the palindrome. Palindrome basically means that when you read it in the forward directions and also the backward directions, they are the same. You realize that? GAA-TTC, when you read it reverse way, also GAA-TTC. Very special. Until now, we do not know why they can do this, but they recognize this what we call the restriction site, which is they have, which on these restriction sites, they have this what we call the palindromic sequence. So GAA-TTC. Okay. So this what it means, this palindromic sequence. When talk about palindromic sequence, basically, when you read it in the forward directions, okay, it match with the complementary strand. So like what I say, that's first you have the GAA-TTC in the forward directions. When you read it in backward direction, also GAA-TTC. Do you realize that? So this kind of sequence is known as a palindromic sequence. So this sequence will be recognized by a particular restriction enzyme, and then what they're going to do, they're going to cleave it. So what they break, in this case, they break the phosphodiester bond. Are you clear? So they're going to break the phosphodiester bond between G and A, for example, G and A. So making it become 2 fragments. So it means that this is a DNA, so it will become like this. Here's G, so CTTAA. It become 2 fragments now. So here you have AATTCG and so on. So it makes the DNA, or it cut the DNA into the fragments. So one example here I show you is the EcoR1. So now don't worry about the enzymes, okay? What are the name of this enzyme? Later I will show you 2 that probably you need to know. Okay, so in this case we have the ECO. ECO stand for what? E stand for the ECO stand for Escherichia coli. It's the E. coli that we always learn. R1 is a restriction enzyme types 1. So what is this EcoR1 actually detect? They detect GAA-TTC. So means they're going to cleave it here. Can you see that? The equal R1. So making it become fragments. So when it cleave it, just now I say that, if it cleave it and they form something like this, correct or not? And this we call as an overhang. So means unpaired. Correct or not? Okay, they cleave. So this kind of end we call as a sticky end. I will explain why we call it as a sticky end. Can you see that? Sticky end. So they have this unpaired nucleotide sequence there. Are you clear? Or we call it as an overhang. Another kinds of the restriction enzyme, when they cut it, they cut it straight. So they produce what we call that no overhang. So this kind of end we call as a blunt end. Not sticky end, but it's the blunt end. Okay, later I will show you the list of the example of some of these restriction enzymes. Okay, so you can see that depending on where and how the restriction enzyme cleave, so it can produce sticky ends or blunt ends. So what's meant by sticky ends? They produce what we call single-stranded DNA overhang. So you can see that in this case, we cut it between G and A. What we're going to have here is the sticky end. Now, why we call it a sticky end? Because when they are unpaired, they can stick back. Why? A and T, they can form hydrogen bonds. A and T form hydrogen bond. T and A, T and A. So it means that it's more efficient in terms of the ligation later. When you want to join two pieces of the DNA together, then this one better. Why? Sticky end. The hydrogen bond can help us to hold two, all right? And then we put the glue. You try to imagine you had two rods, for example. You want to stick them together. Okay, you want to apply the glue. So not so good. But if you make the rod something like this, okay, then apply the glue. So better, right? Correct not? Because the shape complementary, that means base pair complementary. So that's why you can stick them better. So this kind of end is called blunt end. The efficiency is not so good. This one is called sticky end. So it means that when you glue them together, when you paste them together, then the efficiency will be better. So sticky end basically more effective, but they do have some problem. Later we're going to discuss about this. Okay, so blunt end. So blunt end basically they don't produce any overhang. So the blunt end both terminate in the base pair. So in this case, there's a small one enzyme. Small one enzyme recognize CCC, GGG. So they cleave directly straight like this between C and G. Therefore, they produce what we call the no overhang or the blunt end. So the blunt end basically significantly lower. Can I see that? So the effectiveness, the efficiency definitely lower, because just now I say that if you want to glue two ends together like this, no one hold them. So the process, the effectiveness of the process will be low compared those with the sticky end. Okay. So this one again, you do not need to remember, just for your informations only, this one. So guys, when you look at my notes, when I put it as this kind of box, did you know? Okay, so this one actually is just for your informations, not in the syllabus. Okay, only two. Okay, if you want to remember these two, BAM H1, we're going to use it later in one of these what we call this gene transfer as a model. Another one is eco R1. We're going to talk about it. Eco R1, BAM H1, these two, we are frequently use it. What I'm going to show here is, you can see that sticky end productions compared to the blunt end. Can I see that? So small ones, scald ones, and CFU ones, okay, they produce blunt end. Okay, PSD1, TEC1, HIN3, okay, BAM H1, Eco R1, they produce what we call the sticky end. Okay, they produce sticky end. So this you need to know. Okay, what are differences between the sticky end and also the blunt end? So I started with example of the restriction enzyme. So BAM H1, Eco R1, just now I've shown to you guys. Can you see that? They produce sticky end. Then we do have the small one and scald one. Small one and scald one, basically they produce blunt end. Okay. So what is the characteristic here? So a sticky end has a short region of nucleotide that is unpaired. So this unpaired region is called as overhang. Blunt ends, so all the nucleotides are already paired between two strands of DNA at the blunt end. So it means that no overhang. So in this case, complementary base pairing between the end of the target DNA and the end of the vector. So it requires complementary bases at the end so that the complementary base pair take place between the sticky ends of the foreign DNA and the sticky end of the vector. In this case, it does not require. It does not require. Okay. So how about this selection and registration enzymes? In this case, because we want to produce complementary bases, so therefore we need to use the same registration enzyme for that particular end that you want to join them together. Later I will show you. Okay, later I will show you. Okay, but in this case we can use different registration enzymes, okay, to cleave both foreign DNA and also the vector. Okay, so issues of orientation and targets during the ligation. So if you use a sticky end, we can overcome the disadvantage of the potentially inserting the cleaved foreign DNA in the opposite directions. But blunt ends, no choice. We have no way to stop this. Okay. In terms of efficiency of ligations, so more efficient compared to the blunt ends. And for the blunt ends, there's less efficiency when you want to join both pieces together. So I'm very sure that most of you now confused about this part. 1, 2, 3, and 4. So take a 5-minute breaks, then come back. I will use animations to show to you guys what it means by this complementary base pairing between the end of the targets of DNA. Means that the DNA that we want to insert into the into the vector. Okay, and how we select the registration enzyme, and also what it means by orientations of the targets during ligations. Okay, so don't worry at this moment. I will go through these 3 and number 4 with you guys slightly later. Okay, so for the I'll take a break first, then come back for this. Okay, so you can take 5-minute breaks.